no problem. So I will immediately move to a concrete example of how interoperability is handled in a scientific community, namely the plant community. And we will listen now to Cyril Pommier, who is the deputy leader of the Information System and Data Integration team at the URGI INRAE laboratory in Versailles. He's also one of the leads of the plant community in Elixir, and he will present us in 15 minutes the exchange standards and fair concepts for the plant community. Cyril, thank you, and the floor is yours.
Thank you very much for the introduction and for the invitation. And I will give you a slight idea of how we are trying to handle the problematic of data standardization in the plant research community, both led by the European infrastructure such as Elixir and Emphasis, which is plant phenotyping infrastructure, and also in close collaboration with global community and global networks. So what we are trying to do here is simply about data integration, about being able to link environment data, phenomic, with all omics data, genetics, genomics, etc., to be able to do either plant breeding, in other words, to study genetic variation by traits, or to do climate change studies. In other words, it's about studying genotype by environment. And we need to be able to do all of this integration in a fair way. The challenge here is that in each of those three silos of data, you've got different level of data management and data standards available. Indeed, phenomics data is mostly dispersed, and it will remain dispersed. It is highly heterogeneous, very creative. It's not very standardized, but it's getting better and better year after year. Likewise, environment data is highly dispersed and heterogeneous. Genetics, genomics, omics data in general are mostly centralized in reference data repositories such as EBI is one, and the data itself is mostly homogeneous, but there are definitely very important differences within the metadata, which are really heterogeneous. So to address this challenge, to close the gaps, to build bridges between the silos, we need to build interoperability in international networks to make it valuable. In other words, to elaborate on national networks, infrastructures, research institutes that gathers in European networks such as Elixir and Emphasis, with a link to global networks such as the WIT initiative on the CGIRs to build international standards, MyAPI, crop ontology, breeding API, MCPD, with the help of the Research Data Alliance, among others. So first thing, when we are speaking about data standards, what are we really speaking about? There are in fact three types of standards. The first one is about semantic. It's the one that is the most biologist driven and the most easy to understand for plant researchers. It's about describing the data, building controlled vocabularies, in other words, term name and definitions, and to link them when we formalize them as ontologies. The second level is about building structure standards. It's formatting and organizing the data and building the data models to understand it. And in fact, I should say both data models and metadata models. Those standards are plain old CSV, VCF, GFF, or OMIC data, MyAPIs that I will describe later. The important thing I'd like to point here is that to make those standards efficiently built, there needs to be both biologist and computer scientist driven. And finally, we've got technical standards, which provide really data integration and sharing mean and interoperability of tools and systems. And they can be APIs such as GA4GH, for genomic data, or the breeding API, which is MyAPI implementation. And of course, this is really the realm of computer scientists. To glue all of those levels, the key element is the use of persistent unique identifier. Philippe Roccassera just points this in his presentation right before, that using efficient POI are really very important. And we need this not only to identify things such as genes or plant accessions, biological material, but also trait, etc. And for this, we rely on systems such as DOI, URI, and so on. If I try to describe things a little bit more for semantic standards, it's ontologies, and it's quite obvious because it's about annotating one object, protein, a gene, like here a protein from an eprot, with one atomic concept, a protein function, a cellular localization, etc. And the list can be rather long, as we can see here. For phenomic, things can be much more funny. Indeed, when we want to deal with phenotypic traits description or environment trait description, we are not dealing with atomic concept, but with triplets, which are made of one trait, one method, and one scale or unit. The trait, for instance, can be the plant height. The method can be the total height, the height from the ground to the first tassel branch, the last expanded leaf, the youngest growing leaf, etc., etc. And also, it can be the highest pixel corresponding to a plant if you are dealing with high throughput phenotyping. The unit, finally, can be made accordingly, either with international system units, such here, or simply a pixel. As a consequence, if you want to build ontologies of all of those combinations of trait, method, scale, you will end up with real challenge and problem because there is an uncountable number of combinations. Therefore, here, the standard building challenge lies maybe elsewhere in elaborating means to build correctly those traits, those triplets. The plant structure standard now, it's MyAPI, MyAPI being the minimal information about plant phenotyping experiments. Historically, it's about plant phenotyping, but as you will see, it has been highly extended. All the information are available on myapi.org. It has been built by many stakeholders, Elixir and Phasis, Bioinformatics, the CGIAs, etc. It's an open community with requests for comments, GitHub, mailing list, every means that can be used to make a community live. It has many publications. The last one is here in last year from the New Phytologist. I'd like just to underline that the means that we have selected to make this biologist-friendly, researcher-friendly is to build it as a minimum information for biological and biomedical investigation standards. Therefore, we have a target on clear definitions, clear examples. We are proposing the use of Excel templates because when researchers want to handle their data, it's quite easy, and we are doing a lot of trainings. Thanks to this, we've got the possibility to provide an understandable, minimal, and sufficient list of metadata to understand the objective of an experiment, the person involved, what were the procedures, the standard operating procedures, the protocol, which is highly important, and they were highly related to the traits that I just presented, and also what was the biological material experimented. To finish on MyAPI, as I said, it is also computer scientist friendly. And for this, we have made quite a good collaboration with the ISA teams and the breeding API teams to build an explicit data model compatible with ISA tools and the breeding API, as well as validation framework and toolbox built within the frame of Elixir. Keep in mind that MyAPI is a specification which has multiple implementation currently in databases such as GNPIS at INRA and phases information systems such as FIS or PIPA, etc. It has also semantic implementation with the plant phenotyping experiment ontology available on the Agro portal, web service implementation, the breeding API, and file archive through ISATA. And I'm more and more thinking that we should consider bringing MyAPI into Aero Crate, but that's another discussion. To finish with this overview of the different type of standards, the phenotype technical standards is a breeding API. In fact, it goes beyond phenotype because it handles some omic data. Once again, it's an international collaboration. Everything is available on brapi.org. It's a web service specification, which has been initiated by the Excellence in Breeding platform of the CGIAR. It's mainly targeting breeding, as implied by the name, but it can really go much beyond breeding. And INRA and Elixir, mainly Elixir, has done a lot of contribution for the phenotyping specifications. And there are at least one paper if you want to get more information. So if we get back to our initial problem, how do we integrate between those three silos here? For this, we need to go in a little bit more details on what exactly we are dealing with. For phenomics, we know that we've got field experiment. It could be greenhouses, it could be a lot of control chambers or other things. Let's keep it simple for now. It's a phenotyping experiment that produces data tables or images or a lot of other things. And the key elements here are both the location and the biological material. The location, of course, is absolutely interesting when we want to bridge to environment, because it has also data table which are located. Likewise, genetics and omics in general data are produced through omics experiment, of course, and include reads and sequence files and so on, and they are taken from samples which are characterized by biological material. Therefore, to be able to build those blue bridge, we need to identify and to really trigger interoperability through interoperability pivot, key shared resources between all of those silos. And the means that we have selected is to put the same standard, in fact, in all of those silos. This is why MyAPI has been extended beyond its initial scope of plant phenotyping. And to give you an idea of how we have...
You have 2 minutes left.
Perfect. To show you quickly how we have done this for bridging omic to phenomic data. For biological material, it was around the plant material. So around defining the source of the material used, both by setting up persistent unique identifiers for computer science, but also human-friendly identifiers and names. They can be rather fuzzy, but they are really important because those are the ones that are making sense for researchers. And they include infra-specific names such as varieties, chasselas, muscat, Riesling, and so on for vitis, for instance. What I'm also stressing is that we need to be able to store and to trace specific identifiers, synonym, project IDs, whatever will end up being usable as another interoperability pivot among datasets. The second level that we have added in all of the silos is the biological material used for the study, the seed lot identifiers, the cutting identifiers, etc., or the GPS location for in situ plants. And also, of course, the plant samples, traceability, sample processing, and for interoperability means the trace of the samples ID in biosample databases and plant IDs, tracing the origin of the sample. This has been used in Elixir data deposition recommendations and more specifically in the biosample database, where MyAPI has been brought to enable interoperability between ENA, IDVA, and repositories for phenotypic information. Thanks to this, we've got shared data standard between all of those databases with phenotyping DB, which are also accessible through the breeding API, and that will be shortly accessible through Bioschema. And all of this can be integrated either in datasets or in search engines such as Feeder, available as this URL. I like to thank a lot of people. I will not name everybody, just I'd like to salute the Elixir, both plant communities and platforms and colleagues, the agent project, the MyAPI community, the breeding API, the crop ontology colleagues, as well as the Anphasis colleagues. Thank you.
Thank you, Cyril. Of course, the domain is very vast and you could not mention everything, but it was very clear, and I think it is a good thing to advertise for all these efforts in the plant community, and maybe people attending this mini symposium will ask you questions and join maybe the efforts. But unfortunately, we have no time for the questions now, but it will be addressed maybe at the round table, we will see.